Hi, I'm Marike, and together with Jan Stevens and Bart Bruinings and a lot of other wonderful people, uh, I created Bentopy. And Bentopy is a really interesting tool and workflow that we developed to support the assembly and creation of really large and complex molecular models, but it's also really useful generally as a system setup tool. And our article about Bentopy and the workflows it supports, and also a number of very interesting example systems that we created using Bentopy, was just published in Protein Science. And we also created a wonderful tutorial already over the summer that people at the Martini workshop were able to do. And we have now updated this tutorial with a lot of really wonderful recent additions and features for Bentopy. And I'm very excited to show that tutorial as a way of introducing this really cool tool I care a lot about to everybody. So let's let's go ahead and look at this tutorial. So here we are at my workstation, and we can start now by going to the Martini website and find our tutorial. So let's go there, and then you go to tutorials, and then we go to the hands and tutorials section, and then right here we have the Bentopy tutorial. And here you can see it. I want to increase the font size a little bit because otherwise it will be unmanageable. This should probably be good. Yeah, so here you can see this really cool logo I made at some point quite a while back. There's actually a link here to a thread on like a social media post of mine describing how I actually made it, which is an interesting story. And yeah, so it's the Bentopy logo actually packed with a lot of proteins using Bentopy. And here in this tutorial, we'll create a couple of systems, right? So this is not the system we're going to create, but this is one of the systems we highlight in the paper. This is truly just a technical demo, right? But it looks really impressive, and we're really happy that we're able to do this within the span of minutes, which is incredible. And here we'll see that we have like three parts of this tutorial, right? So first we'll just fill a periodic box with a bunch of lysozymes, the classic lysozyme starter protein, you know, for MD. And so we'll fill this box with protein, and then we'll solvate it and look at it, see what we made. And here you can see that we will move on to tutorial 2, and that involves a membrane. So now we need to explain to Bentopy that it should avoid putting things at the membrane, right? We need to avoid collisions when packing. And we can also see here that this blue protein, that's actually a ubiquitin that we're going to be putting in, sits within some distance very close to that membrane. So I'll also show you how to do that. And here you can see a much more involved example that's, I think, very interesting, where we can actually distinguish two compartments in this double bilayer system. And it allows us to, like Bentopy allows us to select one compartment for one kind of thing, for example, and now we put the lysozyme right in that one outside. I mean, compartments are the same, but and select ubiquitin for the inside one, then we still apply that rule, right, for it to sit within that distance. So I'll show you how we can make that. So in order to actually start, we first need to install Bentopy. So here I prepared a shell where we can work, and I will first create a virtual environment. And we call it .venv. And now you can source the venv, and right, so now we have our own new, fresh, clean Python environment, and we can pip install Bentopy. I mean, you'll see in my history, right, in my history of my shell that I've done this quite a lot of times. For Bentopy, we actually have pre-built binaries for Linux, which is fantastic. And yeah, so there we are. And now we can actually move on to downloading the tutorial files. So this tutorial is about building a Martini system. I mean, in our lab, we build Martini systems a lot of the time, but Bentopy actually is force field agnostic. So it doesn't matter whether you're doing an atomistic or a coarse-grained or supra coarse-grained, it could actually handle all of that. Yeah, so now we can get into this and start to download these tutorial files, and then we'll extract them, and then we'll change directory into the tutorial files directory. So there we are. It's downloading. Something weird is happening, but it's fine. Okay, so now we can take a look at what's in here. So we can see that we have MDP files. We'll use that right at the end when we're running a simulation. And then we have our structures directory, which contains a bunch of starting structures. And we also have our topology directory, which contains the topology files for these actual structures. So let's look at the structures directory. So we have a double membrane model, we have a lysozyme model, we have a membrane, single membrane model, and then we have the ubiquitin. So the lysozyme and the ubiquitin are the things that we're going to pack into our space. And then these models constitute these things we already have, right? So these kind of represent things that you would build with other tools that you can then incorporate using Bentopy. So for the topology, we're actually looking at this. So here we see a topology for the lysozyme and the ubiquitin, and then for the phospholipids in these membranes, we also have an ITP and generally just the Martini 3 force field. All right, so we're up to date with that. Now we can actually move on to the tutorial again and see what we can do. So we'll use VMD to visualize some of these things, but first we can create our first input file. So Bentopy works with these different steps, which you don't need to do in this particular order, but they generally appear in this order, right? So this is just a helpful figure. So what we generally tend to do first, but this is only if you have something that you want to pack around or inside of, we mask the structure. We'll get to this later. Then you use Bentopy pack in order to actually determine what positions for all of these input structures, all of these input segments, you actually are actually valid positions. Then we make a nice molecular dynamics coordinate file out of that. Then we can merge it with whatever existing structure we had, if applicable, and then we can finally solvate that. We also have a really excellent solvation tool. So that's what we'll try to do, right? So here we can now finally start with the actual tutorial. And first we're going to make this proteins in a box system, right? So no other structures that we're involving, just lysozymes in a box. Super simple. And we have this wonderfully friendly format here for setting up your system. We call that a .bent file. So in our bent file, we will call this one simple packing.bent. You define some general information such as a title, and a seed is really nice because then it allows you to be deterministic over different executions of the program. And here in the space section, we can see that we provide these dimensions. All of the sizes are in nanometers, and also a resolution, which is this voxel size, right? So Bentopy at the back end works with this voxel representation of space, which allows us to pack spaces really efficiently, right? Quite quickly. This size is really excellent for Martini because it's roughly the size of a bead. And then here we see this includes section, and I will show you what this actually ends up doing later. This is not information that is used per se by Bentopy itself, but it allows us to, when we're rendering out the structure and the topology file, this will become these hash includes, right, in your top file. And that allows you to very nicely manage, together with all of your structures, also manage your topology. And then we have our compartments, and this is where it gets interesting. So this is the most basic compartment you can set up. This is the compartment name that always goes first, and then comes some expression that tells Bentopy what the compartment entails, right? What it involves. And in this case, this compartment called system is actually everything. So that's super simple. And here in the segment section, so every segment is kind of a work order for Bentopy to pack something. It's kind of an instruction, like pack this thing called lyse for lysozyme. Pack 650 of them and load the structure from structure/lysozyme.pdb and put all of that in our compartment called system. Here we can actually start doing this already. So now we need to copy this over. So we'll copy this file, and now we will create one. So maybe we call it simple packing, right? And we paste it in here. And now we can save that. Then we can run the Bentopy pack command. So we'll say Bentopy pack simple packing.bent to placements.json. So this placement.json file is really interesting. It contains, rather than a whole coordinate file, it contains instance-based translations and rotations, right? For every instance of something that we place, we know what the structure is, and then we know what the translation and rotation is to get it at the place that Bentopy pack decided it can be packed. So we can run this, and you see it basically finishes instantly. And here, if we actually want to inspect this placements.json, we can look at it like this. So you see that our title and our size and our seed and a bunch of other metadata, including our topology includes, gets put into this JSON file. And it's really nice because it's very machine readable. So you can also add your own tools between this, right? And here you can see that we have one placement or one segment called lyse with the structure at this path, and then you see these batches. So then we have a rotation followed by a position. So rotation followed by a position, rotation followed by a position. And it just repeats that 650 times, and that will make it possible for Bentopy render to actually create a coordinate file out of this. So let's do that. So we can run their placements.json into packed proteins.grow. And now we can actually inspect packed proteins.grow. So I'd actually rather do that here, because then we have some more room. There we are. So here you can see all of our protein. And you can see that it actually crosses over the periodic boundary conditions because we are packing a periodic system. It's possible to turn off the periodicity if you want, but in this case it's very nice for us because we're creating this kind of infinite space of proteins. See, it's pretty dense. We can go a bit denser if we want, but yeah, here you see all of these proteins nicely in martini pink and yellow. Yeah, so that's really excellent. And now we're on to solvating this system. So now we can use Bentopy Solvate, and I will actually show you how to do this. So we use Bentopy Solvate. We take our input system, and then solvated system.grow is our output system. And here we use these substitutes, which I will explain in a minute. We can neutralize the charge, just like with other solvation programs like GrowMax GenIon for placing ions. So what Bentopy Solvate does is basically GMX Solvate and GMX GenIon, but all does it in one step. It's very memory efficient, and it has really fast input-output for reading and writing coordinate files. And it's actually capable of solvating billion atom systems in very reasonable time. And you also see that we refer to a topology. So that's actually something I forgot to do here. We can also write out a topology. Let's write that out. So now if we look at our topol.top, we can see that it included all of these includes that we also saw in the Bent file, remember? And then it gave the title, and it also put in these 650 lysozymes in our topology. So now we have our grow file and our topology, and they're nicely matched, right? So now we can start solvating. So let's use Bentopy solvate. Let's put in the packed proteins.grow. Let's call this solvated system.grow. We give our topology.top, and then what we can also provide are these substitutes. So actually, with a really nice addition I made very recently, you can also say I want Na and Cl as my ions here. And these substitutes mean that when it finds a water residue that it is allowed to place, right? So we'll remove any water residues from this stamped box strategy of placing solvent. And it removes any water residue that collides with the existing structure that we provide here. And from that set of accepted water placements, it will we can instruct it to either do some count of substitutions or some ratio of substitutions. So this would do 10%. That would be a lot. Or some molarity of substitutions. So we generally want 150 millimolar, so 0.15 molar physiological salt concentration, right? We generally do that in MD. And yeah, so here we can provide that, and then we can put a capital M there to make it molar. But actually we do like to use the MS here, which is molarity with respect to solvent quantity rather than to the box volume, because you can imagine that if we fill the whole box with a lot of stuff, that there's actually much less water than the box volume implies, and this will give us kind of the expected result. So this is different behavior right now than GMX GenIon or other solvation tools give you. Right, like this, where we just have the M for molar, it would give you the same behavior, right, based on the box volume. But we like to do this. And yeah, so that should do it if we also neutralize the charge, right? So these lysozymes, they have a -8 charge, if I recall, or +8 charge. And we need to neutralize the charge because we want a charge neutral system. So there we are. And you can see it placed a bunch of waters, and then it also placed a bunch of chlorides to neutralize that positive 8 charge for those lysozymes. And then there's also this like basal sodium and also chloride concentration, right? So this gets stacked up. So now we can actually take a look at our system. So let's look at solvated system.grow. And yeah, so now you can see there's this whole block of water. Maybe we can kind of distinguish these. So let's say this is name W right here, and this is all that's not named W. And now if we turn this off, we can see all of the proteins and ions. And yeah, so you can see that it just puts a lot of them everywhere. Which is very nice, and it was very quick, and all in one step, which may delight a lot of you. It can be quite cumbersome otherwise, especially if you have, so you can also provide multiple substitutes on substitutes on substitutes, which will all just be added together, which is much more convenient than running GenIon over and over and over again. And it's much faster for large systems. It actually gets usable. So now we're able to move on to the next system. So this is indeed what we created, right? We looked at it. I mean, it looks a bit different for us because this is a lot of time went into this visualization, and we are just quickly looking at our system in VMD, but it's really important to look at your systems to verify that they're correct. And I'll leave this as an exercise to the viewer, right? But there's actually other ways of defining these analytical shapes. So you can also define a sphere at some position, for example, the center, or a cuboid with a start and an end point. So that's really helpful for creating really interesting systems. Okay, now we really get into something Very interesting. So if we now look at VMD structures, membrane.grow. So let's take a look at this membrane. Here you see our membrane is really nice and flat. It's already equilibrated, and we'll use this as our kind of starting point for the next tutorial section. And what we want is that we distinguish this area that the membrane occupies from the area outside it. So I'll leave this running, and now we can say Bentopy mask. Let's take the structures membrane.grow, and I want to visualize labels to labels.grow, or what I can also say is b labels.grow. So now it's going to, so we use a library that Bart Bruinings created, and it's really wonderful to work with because it allows you to distinguish this hierarchy of containment. It's really sophisticated, and basically Bentopy mask just gives a command line interface to this amazing library that contains even more features than this. So we're really happy with that. So we here can see that there's this minus compartment, so that's empty, right? Minus here means empty. And there's this filled compartment, so that's 1, so that's the membrane. And now we can actually inspect them because we created the labels.grow. So we'll add a new molecule. We'll load labels.grow. Let's see, so I just showed that as lines. Maybe we can show that as van der Waals spheres. Increase the size a little bit, and then we can make it nice and chalky. There we are. That's wonderful. And now I want to cut this open. Let's see, y smaller than 200 angstroms. So now you can see that we have this green and this magenta kind of label in our file. So this is the -1 compartment, and this is the +1 compartment. And you can see it matches very nicely with where the membrane actually sits. So this gives us that information. So now we can move on and actually write a mask for it. So we do -L for give me a mask with this label, and then we can do 1. So then we select with 1, we select the label we want, and with after the colon comes the file name that we want to associate with that. So that's membrane mask.npz. And it creates that mask for us. It gives us even some volumetric information on it. That's very helpful. And then we can use that to create a new Bent file. So I'll show that right here. Yeah, so we're creating this file called membranepacking.bent. And this you've already seen. We're also adding ubiquitin as an ITP right here. And then we have our membrane. So we load that from the membrane mask.npz. So this is our membrane compartment. And then we want to distinguish basically everything that's not the membrane, because that's where we'll end up placing things. So we create this solvent fraction that's not membrane. We create a special compartment called close to membrane, which is all of the space around but not including the membrane. So that's within 5 nanometers of the membrane, but not including the membrane itself. We place lysozymes again, and we place 300 of them. We give this a tag of lys, and this we give a tag of ubiquitin before, and then we put it in the solvent compartment, all right? And then here, the ubiquitin, we load from ubiquitin.pdb. We place 100 of it, and we place it in the close to membrane compartment. So we can copy this file over here and create membrane packing.bent. Then we'll save that, and then we can say Bentopy pack membrane packing.bent, and then we create placements.json. So you've seen this before. Basically packs in an instant, which is wonderful. And then we can say Bentopy render membrane or placements.json, and we can call it packed proteins.grow. And now what you will see is that if we now load these packed proteins.grow on top of this file, there we are. I probably need to show them as van der Waals spheres. Just deal with it. Let's see. So here's our system. Now it got a bit complicated with everything here, so we can remove the labels. So you can see that these ubiquitins here in pink, they are placed close to the membrane, and then these lysozymes are also placed close to the membrane, but like wherever, right? So we didn't give that a special around treatment. But so as you can see, you can combine different compartments and you can do kind of spatial rules around compartments, which makes for a really powerful system. So now we can use Bentopy merge to create some combined.grow from membrane.grow and packed proteins.grow. And remember how we used Bentopy render, right, to make our topology. So if you now look at our topology, we need to add right here, we need to add our POPC. So let's see how many that was. That's this number of POPCs. So this is now our membrane section, and this is the packed proteins. And now we combine that into combined.grow. Then we can Bentopy solvate again. This is a great recipe for solvation for us, but then we can call this combined.grow. And now if we take a look at that system, maybe just from scratch here, VMD solvated system.grow. You can see our ugly green water. We can actually maybe remove that, right? And maybe also remove our ions. But this is basically that same system. Maybe we can color it by res name again. And then you can see that, right, we still have a nicely solvated system. And it nicely avoids the membrane here. So that's something we can be very happy with. So now we are ready to move on to the next system, which will be most interesting, I think. So this is what we created indeed. So now what I want to do is I want to take a quick look at this VMD structures double membrane.grow. Let's look at this structure. So here you can see that we now have basically two compartments, and because they connect over the periodic boundaries, you can kind of distinguish this outer section. I will call it outer, even though that's kind of incorrect here, because they have the same, they're like equivalent topologically, right? Like this one is not more inner than that one, it's just translated that way. But yeah, so we can call this compartment A and this compartment B, for example. But we definitely see that there's two compartments, and conventional ways for kind of, I don't know, voxelizing a structure into like a map or something would not allow you to do what we're about to do here. So I'm super excited about that. So let's do Bentopy mask on structures double membrane.grow, and let's do the labels.grow thing again. So now we're loading the structure and it's determining what the compartments are. And then we can now load our labels.grow again, like this. And I want to show them as van der Waals spheres, maybe a bit bigger so we get a nice look there. Maybe we can make them chalky. And also I want to cut them. So let's say something like this. Okay, are you ready? So now if we look at this, you can see that the two membranes have a different color, but then why is this like solvent space still all magenta? Because we can clearly see that there's actually two negative numbers here. So these are the two membranes, and these two are the two compartments that are distinguished by the membranes. So what we need to do is we also need to say and name is like -1, so that's this space right here. And then we can also make a space called -2, and then we can maybe give that a different color. And then on top of that, we can also show 1 or 2 as different compartments here. So now we nicely distinguished our -1 and -2 and our +1 and +2 compartments. So now we wanted to create a system where the ubiquitin is lining this part of the membrane, right? And there's no lysozyme in this section in this compartment. And then here there's just lysozyme, no ubiquitin. So we're going to create that. So now we need two masks for the different sections. And we also need a mask for the membrane. So let's take a look here. Yeah, so we're creating a mask called A mask and a mask called B mask and then a membrane mask. And I'll show you how to do that. So we say, we call -1, we call A mask.npz and then -2, we call B mask.npz, and finally we have 1 and 2, that's our membranes, and we call that membrane mask.npz. So that's great. So I just wrote all of them to have the volumetric information here, and now we're ready to create a cool Bent file. So here we have our Bent file for this system, and I'll copy it and then we'll talk about it. Compartment packing.bent. Let's look at this. So we have a title, we have a seed, we have dimensions, we have a resolution, we have this. So this is all the same as before. I mean, the title is different, but like generally it's the same thing. And we also see our compartments and segments section. And here you can see that we're loading mask segment or compartment A from the A mask and compartment B from the B mask, and we load membrane, the compartment called membrane, from the membrane mask, all very obvious. And now we can do the same around trick like we did before. Now we do around 4 nanometers, and we call it membrane neighborhood. And then here we say, hey, but we only want the membrane neighborhood within compartment B. So here we make a compartment combination by combining the membrane neighborhood and compartment B. The only ubiquitons that will get placed, like we define here, will be in that section on the inside. So this is already quite a sophisticated way of building systems, and it's remarkably easy to define them this way. So now we can do Bentopy pack on compartment packing, get our placements, then we do Bentopy render on our placements, and let's create packed proteins.grow, let's create a topology as well. And we can look at our topology. Now we would have to put in, before we merge, right, we would have to put in our lipids as well. So maybe we can do it like this, just run this. And we can also solvate once again. So let's do our solvation. Nicely use our CL, and we like to use M molarity with respect to solvent quantity. And then we have our nicely solvated system. So let's load it up. And there we are. So that's really excellent. And now I want to color, so that's our ubiquitons. Maybe we can give that a nice color. I like to color those in cyan. And then we get our lysozymes so that we can color in, I don't know, lime. And then we also have our PLPC membrane. So maybe we can just color that by name, right? Of course, there's also solvent in this system. So name WNaCl, so that gets us all of the solvent. Maybe we just want to show the ions, maybe make them a bit smaller to have that nice and clearly visible. So this is a really cool system that we made, right? Let's maybe square it up nicely, and then we say rotate. So we need to rotate the x-axis by -90. So now we're looking at it like this, and now we can rotate the y-axis by maybe 45 degrees, and maybe now rotate the x-axis by, I don't know, 30 degrees. Maybe that's a bit much. Like this, and now we have our very cool membrane system. So I guess that it would now be nice to start simulating this, and we can learn how to do that from this here. I will just copy this because this is not a tutorial about how to simulate molecular dynamics simulations. Many people already know that, and if you don't, it's a really cool thing to learn how to do, and there's excellent tutorials on the Martini website, as I showed you. So I want to create a thing called simulate.shell, like this, and then we're going to source GMXRC. And here, so here it's going to load solvated system, etc. So now we can chmod our simulate.shell, and then we can say, hey, let's do this. So now GROMACS will first energy minimize our model, because Bentopy is very conservative in its placement, which means that we can make pretty much a guarantee that there will be no overlaps between molecules. That's a very strong claim we make that allows you to really rest assured when you're building incredibly large models like our mitochondrial tech demo here, right? Or when you build an entire cell like Jan Stevens does, or when you build a whole aerosol model. I mean, in the meantime, we can actually take a look at our wonderful publication here. So for example, this is a model that's really cool. So this is something that my friend Abby worked on. She used Bentopy in order to pack the whole aerosol model, which is also described in a really excellent paper that can be found here. You see all of these different species. So this is actually an atomistic model, right? We've been working with a Martini model, and now we're looking at an atomistic model. You see the kind of the around placement around this virion in this aerosol, and you see all these mucins, and yeah, it's a really quite striking image. And this just packs in a couple of seconds, so we're very proud of that. This is a remarkably large model. If you solvate this, right, if you solvate the entire sphere, it's a billion particle atomistic simulation. So there is our equilibration. And then what we'll see is that it shrinks a little bit because the solvation, of course, means that it will shrink a little bit. Load data into molecule. And then we can say, hey, let's take this eq.ecstasy. And I think we're about to, yeah, there we are. So now we can load this equilibration. We can look at our little movie. So here you can see it wiggle, and not much is happening, right? Because it's just an equilibration, but you can see it compress a little bit. There's like a semi-isotropic pressure coupling here, as is appropriate for this membrane system. So this is how you would go about building a system with Bentopy and then simulating it, in our case with GROMACS. So yeah, thank you for watching this video where we end up making this really cool model, and I hope that it has been useful.